So today we will continue with the next type of secondary structure, that is the beta sheets or the beta pleated sheets. So in the last class we had already studied about the first type of secondary structure in proteins, that is the alpha helix, right? So today we are going to take a look at the beta pleated sheet. Now these beta sheets, as you can see here in the figure on the right, you can see they are formed when two or more polypeptide chains in their fully extended form, they are brought together side by side. You can see this is one polypeptide chain and this is another polypeptide chain. So when they are brought together side by side, so that the regular hydrogen bonds can form between the peptide backbone of the NH group. You can see here this is the NH group, all right, and this one is the carbonyl group of the adjacent polypeptide. So this is the hydrogen bond formed between them. So you can see when they are brought together so that the hydrogen bond can form between the peptide backbone of the NH group of one polypeptide and the carboxyl group of another polypeptide. So you see here many hydrogen bonds are formed, okay, between these two polypeptides. Now the planarity of the peptide bond, it forces the peptide to be pleated. Okay, you can see this the way the arrangement here. So it is pleated, and the side chains of amino acids protruding above and below the sheet. You can see here this is, let me show you again here. You can see, suppose this is one sheet. Okay, this is the first sheet here, one sheet. This is the second sheet, then the third sheet. So it's like the when you fold a paper, after you open it, it will look like this. So just like folding a paper and then open it again. So the way these beta sheets are formed, that is why they are referred to as beta pleated sheets, because they are formed in this way. And you can see these are groups, okay, this one, this one. You can see one above the sheet, one R group below the sheet. So these R groups, or the side chains of the amino acid, they protrude above and below the sheet. You can see in this first sheet here also, these are the side chain above and this is the side chain below. So all these sheets, okay, in all these sheets you can see the R group of the side chains, they protrude above and below the sheets. Now here the polypeptide, I will show you another picture. Okay, this is the beta sheet. Now the polypeptide chain within a beta sheet is fully extended such that there is a distance of about 3.5 angstrom, okay, from this one alpha carbon atom here to the next alpha carbon atom here. So the distance between each these alpha carbon, just remember it is about 0.35 nanometer or 3.5 angstrom. And as you can see in this figure, that when the adjacent polypeptide chain in the beta pleated sheet, they can be either parallel, as in this case I am showing you the parallel beta sheets, okay. They can be either parallel or anti-parallel depending on whether they run in the same direction, as you can see here in this figure, from the C terminal to the N terminal. This another polypeptide chain also, it runs from C to N terminal. So they are called parallel beta sheets. But when one beta sheet is from C terminal to N terminal, and suppose the other one is from N to C, then it will be referred to as anti-parallel strands. So we can have a mixture of two, okay. These beta sheets, they can be either parallel or anti-parallel. And these purely parallel beta sheets, they are least frequent in protein structure. Just remember, the parallel sheets, they are least frequent, whereas the purely anti-parallel sheets, that is the one which run from C to N, and the other one, it runs from the N terminal to the C terminal. The anti-parallel, they are the most common in protein structure, and these anti-parallel sheets, they often consist of just 2 to 3 strands. Okay, here I'm showing you only two, but it can be 3 strands also. So in anti-parallel, just remember, we have either 2 strands or 3 strands, whereas parallel sheets, they have at least 3 to 15 strands. So around 15 strands here are arranged. Okay, 3 to 15 strands of parallel beta sheets are arranged. And the best known beta sheet structure is the beta keratin. And beta keratin, you know, it is found where? It is found in hair, and also it is found in silk. And this is another diagram. Here you can see figure A, this one on top. It shows the sheets which run in a parallel direction. Why? Sorry, it is anti-parallel, because you can see here one strand is from C terminal to N terminal, whereas the other strand, it starts from the N terminal and it ends at the C terminal. Whereas if you see in the figure below, these are parallel strands. Why? Because both they start from the C terminal, alright, and they end at the N terminal. So just remember that these type of strands, the parallel strands, they have at least 3 to 15 strands arranged. Okay, whereas anti-parallel, we can have only 2 to 3 strands. Now the next type of secondary structure is beta bends. Okay, beta bends, or also known as beta turns. So why these secondary structures are observed in the proteins? In order to fold tightly into a compact shape of a globular protein, the polypeptide chain often reverses direction. Okay, it is not a straight chain, as you can see here. Let me show you. Okay, as you can see here, this is a polypeptide chain, but it is not straight. As you can see, it bends like this. All right, even here. So in order to fold into a compact structure, the polypeptide in a structure, in a secondary or tertiary structure, the polypeptide, it often reverses direction in this way. Okay, making a hairpin or a beta turn or beta bend. Okay, because it's bending like this, it is referred to as a beta turn or beta bend. Now these beta bends, they are commonly observed when a polypeptide chain is required to fold itself into a compact globular form, okay, by changing directions of the polypeptide chain. And in this structure, in this beta bend or beta turn, you can see a tight loop is formed. Okay, this is like a loop which is formed when the carbonyl group, what do you see here? You see the carbonyl group of one amino acid, okay, it is bonded to the amino group of another amino acid through hydrogen bond. So the bond formed between these two is hydrogen bond. Even in this case, you can see the carbonyl group of one amino acid with the amino group of another, of the fourth amino acid. Remember this, of the fourth amino acid. You can see one, this is number 1, this is number 2, 3 and 4. So the amino group of the fourth amino acid along the polypeptide chain, thus resulting in a so-called beta bend reverses. Alright, the resulting so-called beta bend, it reverses the direction of the polypeptide chain. Now suppose if the direction of a polypeptide chain is if the direction of the polypeptide chain is this way, all right, it is this way. Now when it bends, so the direction it changes this way. So in this way, due to the formation of these structures, the direction of the polypeptide chain, it reverses, it changes. And here in these structures, amino acids like glycine and proline, they occur frequently in beta bends. Okay, they usually occur frequently in beta bends. Why? Because glycine, because of its small, the proline, because it has a very small size, and glycine being more conformationally flexible than other amino acids. So beta bends, these two are often frequently found in beta bends and beta turns, and these structures, these beta turns, they are often found connecting the ends of anti-parallel beta pleated sheets and also connecting other secondary structures. Let me show you here. As you can see here, the first structure is the alpha helix, right? So this is the alpha helix. The first structure here is the alpha helix. The second and the third and the fourth structure, they are the beta sheets, which are anti-parallel because they run in opposite directions. So I hope you can see this first structure, which is the alpha helix, and these three are the beta pleated sheets. So how they are connected, you see here, this structure here is the beta bend or the beta turn. You can see this one also. Here also it should be like this. Here, okay, it is connecting like this one and this one. So you see these beta bends or beta turns, they are often found in connecting the ends of the anti-parallel beta pleated sheets or connecting the alpha helix with the beta sheets. So they are often found in connecting the two secondary structures. So next here we are going to talk about the another level of secondary structures. Okay, they are still secondary structures, but they are referred to as supersecondary structures. Now supersecondary structure in proteins, they refer to the formation of a higher level of secondary structure. Okay, you have studied three types: alpha helix, beta turns, and beta sheets. Okay, so this is a higher level of secondary structure with related to alpha and beta structures. Now simple combinations of few secondary structures with specific arrangement have been found to occur frequently in protein molecules. Okay, for example, as you can see in this figure, I'm showing you two alpha helix joined together. So since alpha helix is a secondary structure, so combination of these two alpha helix is referred to as supersecondary structure, or it can be beta sheets, or it can be beta sheet, alpha helix, beta sheet. Okay, so these kind of supersecondary structures, they are referred to as the motifs. Okay, so these units, they have been called supersecondary structures or motifs. Okay, so supersecondary structures, they are seen to occur in many unrelated globular proteins, and also some of these motifs, they are associated with particular function. For example, there are proteins which are involved in DNA bindings, whereas some others, they have no specific biological function alone, but they are part of a larger structural and functional assemblies of a larger protein. Okay, and these, as I had mentioned, they are of different types. Now the first one here is referred to as alpha alpha motifs. Why? Because as you can see here, there are two alpha helix joined together through this beta bend or beta turn here. So you can see The simplest supersecondary structure or motif with a specific function, it consists of two alpha helices joined by a loop region. Now let us see this here. In this case, you can see this is a loop region where these two alpha helix, this one and this one, they are joined together through this loop, and these, they constitute the alpha alpha motif or helix loop helix. Okay, or they are also known as helix turn helix motif. So these are different names, okay, of this motif. And the two successive anti-parallel alpha helices, they are joined by a loop region. They are packed against each other with their axis inclined. So you can see here these cylindrical structures are drawn just to show that the axis of these alpha helices, they are inclined in such a way so as to permit energetically favorable uh, intermeshing of their side chains. Okay, so to permit the energetically favorable structure. And these structure, the alpha alpha motif, helix motif, they are often found in DNA, regulatory proteins, and also in calcium binding proteins. And such structures, they are stabilized, they are also stabilized by the coil conformation of the alpha keratin, okay, in these regulatory proteins and calcium binding proteins. Now coming to the next, the second one, the next motif is beta beta unit, also known as beta hairpin motif. Okay, it is also known as beta hairpin motif. And this simple motif, it consists of two adjacent anti-parallel beta sheets. Okay, in this case you can see three anti-parallel beta sheets formed by sequential segments of the polypeptide chains, and they are relatively connected by a tight reverse turn again here, that is a beta turn or a beta bend. They are connected through this turn, and this motif is often referred to as the hairpin B motif or the beta beta unit, and these, they are often found in proteins like snake venom. The next level of protein structure is tertiary structure. Now, the term tertiary structure, it actually refers to the entire three-dimensional conformation of a polypeptide. So as you can see here in figure C, okay, from first figure here you can see a long chain of amino acids. That is a primary protein structure, which consists of a sequence of a chain of amino acids. And in the next figure, you see the alpha helix and the beta pleated sheets. Okay, these are the secondary protein structure, but in tertiary protein structure you can see the combination of these helices, the pleated sheets, the turns, the beta bends. Okay, they are all joined together, they fold together to form a three-dimensional structure of a protein. Okay, a three-dimensional conformation. Now, the term tertiary structure, it refers to the entire three-dimensional conformation of a polypeptide. And here it indicates that in three-dimensional space, how these secondary structures such as helices, the beta sheets, the beta bends, the turns, the loops, how these secondary structures, they assemble to form domains. Okay, and how these domains are related together. So when these secondary structures, that is the helices, the sheets, the bends, turns, loops, all these, they folded together, okay, they assemble to form a domain. So this here in this figure C, you can see here this is called a domain. Now what is a domain? A domain is a section of a protein structure which is sufficient to perform a particular chemical or physical work, okay, which is able to perform a function, is referred to as a domain. So this whole structure here is a domain. It is formed by the folding of these secondary structures together, the sheets, turns, helices, loops. Okay. Now, here that means and also one more thing you have to remember, in this tertiary structure, these foldings, they are stabilized by electrostatic forces, van der Waals forces, and other forces like hydrophobic interactions, then hydrogen bonds and the disulfide bonds. For example, in the case of cysteine molecules, we see that the disulfide bonds are formed between the cysteine molecules. Okay, so many forces they many of these forces, they stabilize the tertiary structure. Okay, and in this case, in this tertiary structure, you see that the way this structure they fold, okay, the folding, it places those non-polar hydrophobic R groups. Remember, girls, we have studied in amino acids the non-polar hydrophobic R groups, they are buried into the interior, okay, of the structure, whereas the hydrophilic, that is the water-loving residues, amino acid residues, they face the exterior. So in this way, it makes the protein compatible with the aqueous environment in the cell, because we know that the environment in the cell is aqueous, okay, aqueous solution. So to favor to the protein, it is in order to make the protein compatible with the aqueous environment in the cell, this folding, it occurs, this tertiary folding, it occurs in such a way so that the hydrophilic amino acid residue, they face the exterior, whereas the hydrophobic R groups, they are buried in the interior of the structure. You can see here also the tertiary structure, the three-dimensional structure. You can see many anti-parallel or parallel beta sheets, then alpha helices, the beta turns, the loops. All these, they fold together to form a tertiary structure. Now coming to the last level, okay, of protein structure, that is quaternary structure. Now, like I had mentioned earlier, that there are some proteins, okay, such as hemoglobin. If you remember, hemoglobin, it has four polypeptides in its structure. So such proteins containing more than one polypeptide chain, okay, containing more than one polypeptide chain, such as hemoglobin, which contains four polypeptide chain, they exhibit a fourth level of protein structure called the quaternary structure. As you can see in this figure, here in the quaternary protein structure, you can see two polypeptide chain, one in light blue color and one in dark blue color, indicating there are two polypeptide chains. So this is a quaternary structure. Whereas tertiary structure, it is only a single polypeptide chain, where we see many secondary structures folding together to form a tertiary structure, a three-dimensional structure. But in quaternary, there are more than one polypeptide chain. In this case, you can see, for example, structure of a hemoglobin. You can see this figure here. It is a structure of hemoglobin, this one. Okay. Now here in this case, you can see four different types, one in red color, one in orange, one in green, and one in blue color. So there are four types of polypeptide chain, whereas in tertiary structure we has a single polypeptide in which many of these secondary structures, they fold together to form a tertiary structure. So this is the last level of protein structure. And here in this level, in the quaternary structure, it refers to the spatial arrangement of the polypeptide subunits, okay, the polypeptide chains, and the nature of interactions between them. In this case also, just like in tertiary structure, many forces, they stabilize the tertiary structure. So same thing in quaternary structure also, many interactions are involved, many forces such as electrostatic interaction, then hydrogen bonding, hydrophobic interaction, then disulfide bonds, etc. All these, they stabilize the protein quaternary structure. And one more thing you have to remember, proteins which are composed of 2 or 4 subunits, okay, of 2 subunits only, as in this figure, 2 subunits in this case, they are referred to as dimeric. Okay, di meaning 2. So if they consist of only 2 polypeptide subunits, then they are termed as dimeric. If 4 polypeptide subunits, such as in hemoglobin, then they are termed tetrameric proteins. Okay, tetra meaning 4, so tetrameric proteins. And also another important point to remember is that suppose all the subunits are identical. Suppose in this hemoglobin, if all the 4 structures are identical, or the 4 polypeptides are identical, then they are referred to as homo. Okay, homo tetramer. Tetramer, why tetra? Because there are 4 polypeptide chain, so they will be referred to as homo tetramers. But if these 4 subunits, they are different from each other, then they will be referred to as hetero. Hetero meaning different, so they are termed as hetero tetramers. So it depends, if they are same, then homo; if they are different, then hetero. Okay, and the different subunits of heterooligomers proteins, they typically perform discrete functions. Okay, many different functions. And we know that biological systems are abound with numerous proteins, and these proteins, they are mostly occur in their quaternary structure. One example is ribosome, which is an aggregate of different protein subunits and also RNA. And another example is we can just here only, the one of the main example is hemoglobin, and hemoglobin here is actually a heterotetramer. Why? Because it consists of 2 different subunits, 2 alpha subunits. The one on top here, red in color, shown in red and orange, they are the same, 2 alpha subunits, and below 2 beta subunits. So since it consists of 2 different subunits, 2 alpha and 2 beta, so it is referred to as a heterotetramer. Tetra, why? Because there are 4 subunits. So these are some of the examples of proteins which occur in a quaternary structure.